At the moment of conception, when sperm meets egg, a remarkable journey begins, the development of a new human life. Within this single fertilized cell lies a blueprint, a genetic code carried on chromosomes that determines many of the baby's characteristics, including its biological sex. This code is written in the language of DNA, and among its instructions is the initial determination of gender. Two special chromosomes, X and Y, play a starring role in this process. Each parent contributes one chromosome to the mix. The mother always provides an X chromosome, while the father can contribute either an X or a Y. If the father contributes an X, the baby will have two X chromosomes, XX, and will develop as a girl. If the father contributes a Y chromosome, the baby will have one X and one Y chromosome, XY, and will develop as a boy. This initial genetic blueprint sets the stage for a cascade of hormonal events that will shape the baby's development over the next 9 months. However, assigning gender based solely on chromosomes paints an incomplete picture. While the presence or absence of a Y chromosome establishes the genetic sex, it is the intricate interplay of hormones that orchestrates the physical development of male or female characteristics. Hormones act as messengers, carrying instructions throughout the body, guiding the formation of reproductive organs, the development of secondary sexual characteristics, and even influencing brain development. The journey from chromosome to fully formed baby is a testament to the power and precision of these chemical messengers. The first few weeks following conception are a time of rapid cell division and differentiation. The initial cluster of cells gradually transforms into a recognizable embryo, with the beginnings of organ systems taking shape. During this critical period, genes on the Y chromosome, if present, initiate the production of a hormone called testosterone. This marks the start of the hormonal symphony that will shape the baby's gender. Testosterone, often referred to as the male hormone, plays a key role in the development of male characteristics. Its presence or absence sets in motion a chain of events that will determine the course of sexual differentiation. In a male embryo, testosterone acts as a signal, directing the development of male reproductive organs, including the testes, prostate, and penis. Without testosterone, the embryo will develop along a female pathway, forming ovaries, a uterus, and a vagina. The timing of testosterone production is crucial. It typically begins around the 7th week of gestation, a pivotal point in the developmental timeline. Before this period, the embryo possesses structures that could potentially develop into either male or female genitalia. Testosterone's arrival tips the balance, steering development towards the male trajectory. The absence of testosterone allows these structures to develop into female genitalia. In a male embryo, testosterone acts as the maestro, conducting the development of male characteristics. Its influence extends beyond the formation of reproductive organs, shaping other aspects of male physiology. Testosterone guides the development of the Wolffian ducts, embryonic structures that will eventually give rise to the male reproductive tract. It also promotes the regression of the Mullerian ducts, which would otherwise develop into female reproductive structures. Testosterone's impact is not limited to the physical realm. It also plays a role in shaping the developing brain, influencing its structure and function. Research suggests that testosterone exposure during fetal development can influence the organization of certain brain regions, potentially contributing to sex differences in behavior and cognition. While the precise mechanisms are still being explored, testosterone's influence on brain development underscores its far-reaching effects. The production of testosterone in a male fetus is not a one-time event. It continues throughout gestation, ensuring the continued development of male characteristics. The testes, once formed, become the primary source of testosterone, secreting this vital hormone into the bloodstream. Testosterone levels typically peak during the 2nd trimester, contributing to the rapid growth and development occurring during this stage. In the absence of testosterone, the female developmental pathway unfolds. Without the signal from testosterone, the embryo's undeveloped gonads, the precursors to reproductive organs, differentiate into ovaries. The Mullerian ducts, no longer suppressed by testosterone, flourish, giving rise to the fallopian tubes, uterus, and upper portion of the vagina. The absence of testosterone also influences brain development, shaping neural circuits in a manner distinct from the male pattern. While estrogen, the female hormone, plays a role in female development, Its influence during fetal development is less pronounced than testosterone's impact on male development. The female developmental pathway is often characterized as the default pathway, unfolding in the absence of strong hormonal signals directing development towards a male trajectory. This is not to say that female development is a passive process. The development of ovaries, the intricate structures of the female reproductive tract, and the complex hormonal cycles that will govern a woman's reproductive life are testaments to the intricate choreography of genes and hormones orchestrating female development. The absence of testosterone simply marks the starting point for a different, equally remarkable journey. While the absence of testosterone is the primary driver of female development, other hormones play supporting roles in this intricate narrative. Estrogen, produced by the developing ovaries, contributes to the growth and maturation of the female reproductive tract. It also plays a role in shaping the developing brain, influencing neural circuits involved in female typical behaviors and cognitive functions. Another key player in female development is anti-Müllerian hormone, or AMH, produced by cells within the ovaries. As its name suggests, AMH promotes the regression of the Müllerian ducts in males, preventing the formation of female reproductive structures. In females, AMH levels are typically low, allowing the Müllerian ducts to flourish and develop into the fallopian tubes, uterus, and upper vagina. The interplay of these hormones, alongside the absence of testosterone, ensures the coordinated development of the female reproductive system. Estrogen promotes the growth and maturation of female reproductive organs, while AMH prevents the formation of male structures. This delicate hormonal balance is crucial for the development of a healthy female reproductive system capable of supporting future pregnancies. The development of male or female characteristics is not simply a matter of on or off switches. It is a delicate dance of hormones, with levels fluctuating and interacting throughout gestation. The timing and concentration of hormones are critical, influencing the trajectory of sexual differentiation. Even slight variations in hormone levels can have significant impacts on development. For instance, in males, insufficient testosterone production or disruptions in testosterone signaling can lead to incomplete masculinization, resulting in ambiguous genitalia or other developmental variations. In females, exposure to high levels of testosterone during critical periods can masculinize certain aspects of development, potentially affecting reproductive anatomy, brain development, and behavior. The intricate interplay of hormones highlights the complexity of gender development. It is not a simple binary process, but rather a spectrum of possibilities influenced by a symphony of hormonal signals. Understanding this complexity requires moving beyond simplistic notions of male and female hormones and appreciating the nuanced roles these chemical messengers play in shaping development. The hormonal symphony orchestrating gender development is not always a flawless performance. Variations in hormone levels can occur, leading to a range of developmental outcomes. Sometimes these variations result in intersex conditions, where an individual's physical sex characteristics do not fit typical definitions of male or female. These conditions can involve variations in chromosome combinations, hormone levels, or the responsiveness of tissues to hormones. For example, individuals with complete androgen insensitivity syndrome, or KISE, have XY chromosomes indicating male genetic sex, but their bodies are insensitive to testosterone. As a result, they develop female external genitalia, despite having testes internally. This condition highlights the crucial role of hormone receptors in mediating the effects of hormones. Other intersex conditions, such as congenital adrenal hyperplasia, or CAH, involve overproduction of androgens, hormones that promote male characteristics. In females with CAH, this can lead to masculinization of the external genitalia, despite having XX chromosomes. These variations underscore the delicate balance of hormones required for typical gender development and the diverse ways in which this balance can be disrupted. While genetics and internal hormonal signals play primary roles in gender development, external factors can also influence this process. Exposure to certain environmental chemicals known as endocrine disruptors can interfere with hormonal signaling, potentially affecting sexual differentiation. These chemicals can mimic hormones, block hormone receptors, or alter hormone production, disrupting the delicate hormonal balance crucial for typical development. For instance, exposure to phthalates, chemicals commonly found in plastics and personal care products, has been linked to disruptions in male reproductive development. Studies suggest that prenatal phthalate exposure can interfere with testosterone production and action, potentially contributing to developmental abnormalities in male reproductive organs. Other endocrine disruptors such as bisphenol A or BPA found in some food packaging have also raised concerns about potential impacts on hormonal balance and reproductive development. While more research is needed to fully understand the long-term consequences of endocrine disruptor exposure, these findings underscore the vulnerability of the developing fetus to environmental influences and the importance of minimizing exposure to potentially harmful chemicals. The journey from chromosomally determined sex to a fully formed baby is a testament to the complexity of biological processes. The interplay of genes and hormones orchestrates a delicate dance, shaping the development of male or female characteristics. This process is not a simple binary but rather a spectrum of possibilities, influenced by a symphony of hormonal signals. Understanding this complexity requires moving beyond simplistic notions of male and female hormones. Each hormone plays multiple roles, influencing various aspects of development. Testosterone, while often associated with masculinity, is also essential for bone development in both sexes. Estrogen, often linked to femininity, plays a crucial role in cardiovascular health in both men and women. Appreciating the nuanced roles of hormones allows for a more comprehensive understanding of gender development. It highlights the interconnectedness of biological systems and the delicate balance required for typical development. This knowledge paves the way for more informed approaches to addressing developmental variations and promoting healthy outcomes for all individuals. The intricate dance of hormones during fetal development underscores the spectrum of human sex development. While the binary categories of male and female are useful for many purposes, they do not fully capture the diversity of human biology. Intersex conditions, variations in hormone levels, and the influence of environmental factors highlight the fluidity of sex and gender, challenging rigid classifications. Recognizing this spectrum requires moving beyond simplistic notions of biological sex as solely determined by chromosomes or genitalia. It necessitates acknowledging the multifaceted nature of gender, encompassing biological, psychological, and social dimensions. This broader perspective fosters inclusivity and respect for individuals who may not fit neatly into binary categories. Honoring the spectrum of human sex development promotes a more nuanced understanding of gender and encourages empathy for individuals who may experience gender differently. It challenges societal norms that enforce rigid gender binaries and creates space for diverse gender identities and expressions. Embracing this complexity enriches our understanding of human biology and fosters a more inclusive and equitable society. Subscribe now to join our community. Give a thumb to encourage us to bring more interesting, challenging, and high-quality videos to everyone. Our channel MedSign Health 360 has many more videos to bring the crucial, latest, and systemic medical and health knowledge with a simple approach to everyone for better health and a better life. We'd love to hear your feedback. Share your thoughts in the comments below and let us know what you think.